So, good morning, everybody. We are starting this International Microbiology workshop, and thank you for the presence of everybody. So, here this morning, we have Dr. Jenny Persson from Lund University. She is here with us to talk about the innate response against microbial interactions. Jenny is from Lund University, and she was my supervisor during my PhD. And thank you very much for being here with us today, Jenny. Thank you.
Thank you. Thank you very much for the invitation, Tiago. It's a pleasure really to be here, and I hope that you will enjoy the presentation.
Thank you.
Can I get it started?
Yes.
So, welcome everybody, and again, Tiago, thank you very much for the invitation. So my name is Jenny Persson, and I'm an associate professor at Lund University in Sweden. I currently work at the section for immunology, and my current focus is research focus is mostly on innate sensing of microbes and so how the innate part of the immune system recognizes microbes. And I have a background in medical microbiology, so I'm going to connect some of these sensing pathways that I will be talking to you a little bit about to my favorite bacterium, which is group A Streptococcus or Streptococcus Pyogenes, and that Tiago was also very fortunate to have had to work with when he was here. So I will get started. So just a short background recap of infectious disease. As you know, infections has been with us as all times, basically. And for the most part, they're not making much fuss about themselves, but every now and then something pops up that really creates great disasters in the world. We had, for example, the Black Death during the 14th century, which killed about 100 million people, which at the time was a lot. This was mostly in Europe, but those 100 million people was about 20% of the entire population of the world. So there's a lot of people that were killed by this infection plague and this was caused by a bacterium called Yersinia pestis that spread mostly through rats, as I think you know. And then we have the 1918 flu, which was also mostly in Europe, or the Spanish flu, which spread across Europe after the First World War. And it killed about 50 to 100 million people. It's debated a little bit how many people actually died from the flu and how many people died from secondary bacterial infections. This was caused, the flu was caused by one of the influenza viruses. And an interesting comparison here is that basically this 1918 flu actually killed as many people in 24 weeks as the Black Death did in 24 years. So even though these with a historical aspect, these are, of course, both horrible things that happened, but they have also very different kinetics. So the 1918 flu, of course, was extremely dramatic event. It was very quick. And today we have the AIDS pandemic, which is basically still going on, although AIDS is, at least in many of the industrialized countries, it's a manageable disease to live with. But still, we have about 2 million deaths each year, and we've had about 25 million deaths since the start of the AIDS pandemic. And this is, of course, as you know, caused by the HIV virus. And unfortunately, we are still in the middle of of the most current pandemic of COVID. And now we have about 5 million dead in the world so far, and the pandemic is far from over, it seems like. So hopefully it won't get much worse, but of course we can't know. And as you all know, this is caused by a new coronavirus, which has been named SARS-CoV-2. It's quite interesting to actually look at infectious death across the world, because as you know, most infections that we are aware of, we can treat. And these difficult ones that pop up every now and then, like HIV or now the coronavirus, SARS-CoV, they're the rare exceptions. In general, viruses are more difficult to treat than bacterial infections, but for the most part, we can manage them anyway. But still, these infections are on a global aspect within the top 10 disease-related causes of death. We have lower respiratory infections, we have neonatal infections, and diarrheal diseases. So these are all communicable diseases, and within all the disease causes, deadly causes, these are things that could be treated. And you see these circles here. The open ones is for 2000, and the closed ones for 2019. So at least what you can see is these communicable diseases have become slightly less common. So they are being managed, but of course much more could be done. While these non-communicable diseases like ischemic heart disease and stroke, they are going the other direction, unfortunately. And these are more usually lifestyle dependent diseases. But the communicable diseases are of course caused by pathogens. So what then is a pathogen? We are constantly meeting microbes, bacteria, and viruses and fungi every day, hundreds and thousands of microbes. We eat them, we breathe them in, we touch them, but for the most time we don't get sick. And some of these microbes are pathogens, and they can and sometimes will cause disease. But the question is then, and this is somewhat of an esoteric question, what is a pathogen? What really makes a pathogen a pathogen? And the short version of this is that it's a microbe that is able to cause disease in a normal host. And by normal, I mean not immunocompromised. Because basically any bacterium, if you inject it straight into the blood, for example, of a person, it will cause disease, or at least a very bad inflammatory response that can be lethal. And of course, also in patients where you have deficient immune responses, you will also maybe get disease caused by some bacteria, for example, that normally wouldn't cause disease. And those bacteria are actually usually not called pathogens, although they can cause diseases. So what's also typical for pathogens is that they express what we call virulence factors. And a virulence factor is a component of a pathogen that contributes to disease development in the host. And they can be adhesion molecules, they can be different types of toxins, they can be proteases, they can be sugars, pretty much anything that is produced by the pathogen, and when it's inside of the host, cause something that makes the bacterium or virus pathogenic, i.e. to being able to cause disease. So most pathogens express, or all pathogens express more than one virulence factor. So a multitude of virulence factors that are active at different sites in the body and responding or protecting the bacterium against different things in the human body. And one thing that is important to think about when we talk about infectious disease is that it really is an evolutionary arms race that we as humans can never win. So no matter really what we do in terms of vaccines or treatments, the bacteria or viruses will in the end always outsmart us. So this is not something that we can ever stop working with. I know in in the US in the 30s, or 40s, when antibiotics became a big thing, the Surgeon Colonel General in America at the time said that we have now won the battle against the microbes, which of course we know now was completely wrong and very naive. And one of the reasons for this not basically ever being able to happen is that if you look at the human in this, we have an average generation time of about 30 years. And as a comparison, your average pathogen has a generation time of about 30 minutes. So that means that we won't ever be able to adapt to any given pathogen change immediately when we are sick. Over time, we can, of course, but it will take a long time. In contrast, most pathogens can evolve and change and adapt to things in our bodies very rapidly, including the introduction of a vaccine, for example. So then what does it take to be a successful pathogen? There are several things that all pathogens have to do to be successful. One of them, of course, is to actually get into our bodies, get across the physical barriers, the mucosa, the skin, for example. Once that has been accomplished, they will face the innate immune system, which are several different systems actually in our body that responds very quickly and acutely. Once that is passed, there's our adaptive immune system that also has to be dealt with. And once that has all been taken care of, the last and maybe most important thing is spread. So it's not enough for a bacterium to cause disease in one host, which it would be able to do here with all of these three accomplishments. But to be able to actually be a successful pathogen, it also has to be able to spread within the society. And this is actually a very important aspect of what makes up a pathogen. So if we compare or think about really what is a successful pathogen and how does this relate to what type of or how bad a disease a pathogen creates. So if we think of the term virulence, which is a term that we use to identify how good a pathogen is at causing disease. So it's exactly, it says here, so it's a relative ability of the pathogen to cause disease in a host. And it's important to think also that the disease that is being caused is almost always caused by two main things. And one is the actual tissue destruction, for example, or the breach that is caused by the pathogen. And the second aspect of disease is that breach of tissue homeostasis by the pathogen will create an inflammatory response. And very often that inflammatory response is also part of the disease, of the pathological picture in the patient. But if we go back to what really is disease by a successful pathogen, and how does that relate to virulence? So, for example, if we look at the Ebola virus that was, we had a big outbreak a few years ago in parts of Africa. And this is an extremely virulent pathogen. So meaning that almost everyone that gets this disease, they die. So I think there is an 80% death rate with the Ebola virus. This is a terrible, terrible pathogen. And then somewhere down here, in terms of virulence, if we compare to the Ebola virus, we have the influenza virus that causes, of course, every year, thousands and thousands of infections, and some very serious ones. But for the most part, it's a pretty benign disease. that most of us come out of after a couple of weeks with no major issues. So if we then compare this to what makes a successful pathogen, if we add another axis here, what does that look like? It actually looks like this. So the very, very virulent pathogens are actually pretty bad pathogens, while, and as is the very, very, the pathogens that end up on the very, very low scale of virulence. If you're not good at causing disease, you're not going to be a good pathogen. But if you cause too much disease, you're actually an equally bad pathogen. And the really good pathogens are the ones that cause moderate disease. And that is because of a couple of things, but mostly it has to do with spread. So of course, if you kill off your host too quickly, you won't be able to spread. And I mean you, if you're a microbe here. This is the disease of microbiologists. You always refer to, you see yourself as the microbe in any given occasion. So if you're good at killing your hosts, you're not going to be able to spread to new hosts very well. So this is actually, on the scale of things, a pretty bad idea. And most successful pathogens actually fall within this range, this middle range here. Like the influenza virus, for example, which is a very efficient and very good pathogen. So to sum that up, most successful pathogens are very well adapted to their hosts, meaning that they can cause disease and spread, but they're actually not causing disease that is so bad that spread is broken off. So they are appropriately virulent to establish and maintain infection, to ensure spread within the population. So this ensuring of spread is very important. So if you're a pathogen then, and you have managed to enter somebody's body, you've been able to breach the mucosal surfaces, for example, then you will, as I said, meet the innate immune system and the adaptive immune system. And I will focus today on the parts of the innate immune system, which is my main area research-wise. And innate immunity really is several different systems. And we have cell-based systems of different types of immune cells, innate immune cells. And the things that I will be talking about today are actually mostly referring to macrophages and dendritic cells and to some extent neutrophils. But I'm going to be focusing on some of the molecular aspects of innate immunity in these cells. And then we have something called the complement system, which is a humoral system within innate immunity. And actually both adaptive and innate immunity have both cell-based and humoral parts of their responses. So humoral meaning something that is present in the bodily fluids, like in the blood, for example, or in the plasma. So I will start with the main humoral system of innate immunity, and that's the complement system. So the complement system is a family of about 30 to 40 proteins that are soluble and present in our blood or in the plasma of our blood. It's a proteolytic cascade that can be activated through three different types of pathways. So the first pathway that was identified is now called the classical pathway. And this pathway is dependent on the presence of specific antibodies. So when antibodies has bound to a microbe, for example, this can be recognized by the complement protein C1q, and that then activates the complement cascade. And this cascade is basically a proteolytic activation of different proteins. So C1q activates complement protein C4, which is then cleaved. And then as that becomes active, it activates the next complement protein C2 and so on. And the other two pathways that we have here, one is called the Lectin Pathway, which is dependent on specific lectins that can recognize sugars on bacterial surfaces. An activation of this pathway has the same principle as the classical pathway. So C4 is activated and then C2. The alternative pathway is a little bit different. It is a pathway that is continuously, spontaneously activated. So the key protein for all of these pathways is protein C3. And in the alternative pathway, C3 is activated by a spontaneous takeover mechanism. So there's always a little bit of activation. And if there is a surface present, then C3 that has been cleaved can bind to that surface, and so that pathway can continue through spontaneous activation. And C3 really is the main functional protein of the complement pathway. It's cleaved into something called C3b, and C3b can then be attached to microbial surfaces, which makes them visible for phagocytes. And this is a process known as opsonization. So C3b is deposited onto microbes, and then, for example, macrophages can recognize C3b and eat up the microbe. We also have the production of two small proteins known as anaphylatoxins. They are also cleavage products of C3 and C5. So all three pathways will activate C3, and then that will continue onto activation of C5. And C5a and C3a are very strong anaphylatoxins, meaning that they cause inflammation and they recruit other immune cells to the site of infection, for example. Once C5 has been activated, this can cause the insertion and generation of a big membrane pore in microbial surfaces. And this is called the MAC complex, or the membrane attack complex, and contains a number of C9 molecules together with C5 to 8. And basically, when this happens, it creates a pore in the microbe that creates fluid influx into the microbe, and the microbe will lyse and die. But really the main aspect of the complement system is C3. This part down here is important mostly for certain gram-negative infections, the complex. And for example, if you have deficiencies in any of these late complement proteins, C5 to C9, you are very sensitive to Neisseria meningitis, but almost nothing else. Whereas if you're lacking anything of the activation pathways or C3, you are pretty severely deficient for being able to take care of most common infections. And you have patients like this usually have recurrent difficult infections during childhood. And this system is, as you might understand, tightly controlled. So if we had no control of this system, we would, because of the alternative pathway, have constant activation of complement, which of course would be a pretty bad idea. So that's tightly controlled, and basically at any level, there are several different types of regulators that keeps this under control. Yes. So another aspect of innate.
Jenny.
Yes.
Excuse me. It's just from your last slide.
Yeah.
We got a question, the last one.
Yeah. This one?
Sorry. The other one. Before that.
This one.
Yes. No, the one that we had those lines. We
This
got
one.
yes, this one. Thank you. We got a question from Eloisa.
Yeah.
And she asked if in this case, Ebola virus is not considered a successful pathogen because it kills its hosped very fast.
Yes.
Is that?
That is correct.
Yes. Okay.
So exactly. So the Ebola virus is extremely virulent. So it's a very potent pathogen in that sense. But from an evolutionary perspective, it's not a very successful pathogen because it's very limited in its spread. So we have these rare outbreaks, but as soon as they're controlled, it's shut down. So it's not a pathogen that is very good at spreading within the population. So from an evolutionary point of view, it's not actually considered a very successful pathogen.
Yeah, and do you think it could become like a pandemic from Ebola?
No.
No?
Not under the no. Because it can't spread fast enough to actually create or generate disease in people that travel, for example. So it's to be able to spread, a pathogen needs to spread faster than we can contain it.
Okay.
Which is not the case with Ebola virus at the moment. If the Ebola virus would change, say that it changes in a way that we carry it without having any symptoms for 2 weeks before anything happens, then of course a lot of people will be able to travel and maybe spread the virus before we actually get any symptoms.
Okay.
So that would, for example, change the ability or the successfulness of the pathogen. But luckily, hasn't happened with the Ebola virus yet.
Yeah. Okay. Thank you.
Thank you. Yes, please ask a question as soon as you have them. That'd be great. Okay, where were we? Here. So yes. So now I want to switch focus a little bit and talk about another aspect of innate immunity. So the complement system is a humoral part of innate immunity, and it's present in the fluids in the body. And we also have our cell-based protection lines, and in terms of innate immune cells, and they're, for example, macrophages and dendritic cells and neutrophils. There are a few others as well, mast cells, basophils, and eosinophils. But these receptors that I would like to talk about now, the pattern recognition receptors, are mostly expressed by macrophages, DCs, and neutrophils.
And Jenny, I'm sorry, we get some questions again related to Ebola.
Yeah.
Is that okay?
Yeah, of course.
Yeah. So Anna asked if Ebola is not transmitted by the air. So how is it transmitted? If you don't know that.
So it is, as far as I know, Ebola virus is something that you get usually from eating infected meat. And then once that has happened, it can be spread through the blood. So these patients, of course, they usually bleed a lot. It's a bleeding disease. So they start bleeding from the eyes and from the nose, and all of the mucosal membranes are destroyed. And the blood is infectious. So it's very infectious. And I don't know if you saw during these Ebola virus outbreaks, they tried to put all of these patients in isolated tents, and all of the staff that was in there were completely covered in these space suits. So it's an extremely contagious pathogen. So but it doesn't spread quickly enough, because these patients die.
And another thing from Anna is, like, if I understood her question, is if you believe that to become a pandemic, the pathogen must be airborne, transmissible by air.
Does it have to be airborne? Theoretically, no. But I would guess that most pandemic-causing pathogens will be airborne.
If we think about plague, as you showed before, it's not an airborne disease, and became pandemic.
Yes.
So,
but of course, the ability to spread through air is, of course, a good ability if you want to, if you're a pathogen and want to create a pandemic. But it's not necessarily, it's not necessary.
Okay.
Yeah, and I guess it's also it's a bit tricky to, because of course the Black Death was, of course, it was over many years. So that was the amount of people that killed. That probably wouldn't happen today because the society is very different. So the spread via rats that way that it happened back then, I wouldn't think that it would happen today. Plus, we have antibiotics, so
yes,
we'll be able to treat the bacterial infection. Viruses are trickier because we're not as good at treating them. Most of the viruses that spread now that cause pandemics, like these different influenza viruses that have been spreading, that were spread every year, and also these different coronaviruses that spread, both the current one and the old MERS and the SARS viruses, they are all, as far as I know, spreading via the air.
Yeah. Okay. Thank you. And the last thing is I think it's not a question, but it's from our YouTube channel. It's Que Alquimia é Essa? And it's asking if longer incubation periods without symptoms would contribute to successful spread. So that would be the key.
Yes.
Okay. Thank you.
Or a key at least, or one important aspect. Yes.
Yes. And from your complement part, the one you were showing before, does the complement system just attack bacteria, or it is important against viral infection as well?
It's important against viral infections as well. It's mostly actually, if you look at the diseases or the deficiencies that happen to people that lack proteins of the complement system. So there are certain deficiencies, and in terms of infections, they are usually very sensitive to certain bacterial infections. But it is also activated by by viruses, and especially the classical pathway here, which you see, is actually activated by specific antibodies. So in this case, the complement system won't actually ever know, is this a bacterium or is it a virus?
Okay.
But because most viruses are, when they're in our bodies, they are inside our cells, right? Yes. So they're actually only exposed to complement for a very short period of time. And any viruses that manages to get into our cells won't actually be recognized by the complement system. They won't be accessible. And of course, the viruses have their replication niche inside our cells. So as soon as they've been managing to get into a certain amount of cells, they will be causing disease anyway. And the same with the alternative pathway. So the alternative pathway basically needs a surface, and that can be any surface. It doesn't have to even be a bacterial surface. It can be plastic. But as soon as C3b is attached to a surface, that will create complement activation onto that surface. So that's basically non-specific. And the lectin pathway is pretty bacterial specific. But so the question is, no, not only bacteria.
Okay, thank you.
Thank you. Okay, so pattern recognition receptors. So the pattern recognition receptors, it's a large family of receptors that we have in our most of our innate immune cells. And they are bound to membranes like the plasma membrane or endosomal membranes, or they are present within the cytosol. And these receptors are activated very quickly. So as you can imagine, the complement system really activates or is activated immediately as something comes into the blood. But the cells of our immune system will be present maybe in our tissues. So when something enters a tissue, it's more likely to be sensed immediately by any of the pattern recognition receptors. And this is very quick, and they are expressed by almost all immune cells. What is important to remember, though, is that we have a vast amount of pattern recognition receptors, and not all cells express all receptors. And that gives the cells a little bit different ability to what they are able to respond to and how they're able to respond. Another important aspect of pattern recognition receptors is that they are what we call non-clonal. So in B cells and T cells within the adaptive branch of our immune system, their receptors are developing in a clonal way. So a cell that recognizes a bacterium, for example, and gets activated has a specific receptor that is then being expanded upon. That's not the case for pattern recognition receptors. So they are they're not able to expand in the way that cells from the adaptive immune system are. And what's also specific for these receptors is that they are recognizing microbial structures very specifically, but they can't really recognize very many of them. So they have a quite limited amount of specificities. And they also can't change. So also in contrast to the receptors of the B cells and T cells, which can adapt and be more and more specific, the PRRs are germline encoded, and they won't be changing with an infection. They won't get any better at sensing anything, for example. So what do these receptors then recognize? So most of them or all of them can recognize something that we call PAMPs, or pathogen-associated molecular patterns. So these are specific patterns that are present on microbes. And these PAMPs can be, for example, constituents of bacterial cell wall, like LPS, for example, of the gram-negative bacteria, or lipoproteins, which are present in most gram-positive bacteria. It can be parts of bacterial structures, for example, flagellin, which is the protein that makes up the bacterial flagellum, so that some bacteria uses to swim. Or they can sense genetic material, so RNA or certain kinds of DNA that are present within bacteria or viruses. And what's important to remember about these PAMPs is that they are conserved within each microbe, and they are essential. And this is very important for the function of the PRRs. So because the PRRs cannot change, it's important that what they are recognizing is also not changing. Because if they were changing, then of course we would lose our ability. Remember this generation time difference. If the bacterium or virus change any of these structures, then we wouldn't be able to use our PRRs anymore. So this is of course something that has then evolved over many, many years in the humans, that these receptors are actually targeted to parts of the microbes that are essential. They're not changing, and they are essential. And essential meaning that, for example, LPS, a bacterium that expresses all gram-negative bacteria express LPS. And if you mutate those bacteria to not express LPS, they will die. Same thing, bacteria that uses flagella to move are often dependent on the presence of these flagella for their ability to spread and replicate. So if you remove the flagellin, so they can't make the flagella, they don't survive as well. Same, and here for the genetic material, this is pretty obvious. I mean, if you create a mutated bug, any bug, virus or bacterium, to not contain DNA or RNA, they would obviously not survive. So that is very important for these receptors, the function of these receptors, that they are actually over time have managed to target parts of microbes that the microbes cannot change. So it's very important. PRRs also recognize other types of molecules, or another type of molecules called DAMPs, or damage-associated molecular patterns. And these are proteins usually that we have inside our cells. So when we're not sick or we don't have an inflammation going on or an infection, these proteins are not visible to our immune system. But as soon as our cells start to die, for example, during issues when we have necrotic cell death. So necrosis, as you know, is a type of cell death where the contents of a cell is just released into the environment. And this can happen during infections. Most infections you have necrosis going on, but also during certain types of sterile inflammation. But then our PRRs will recognize some of these proteins that are then released, even though they are our own proteins. And some of them are listed here. So, for example, histones are very potent DAMPs. ATP, which is, of course, an essential molecule for just the general function of a cells. But the extracellular environment usually have very low amounts of ATP. And once we have a lot of cells dying and the ATP is released into the extracellular environment, some of our pattern recognition receptors can recognize ATP and activate an immune response. So these are, as I said, these are self molecules that are basically a sign of damage or danger. And also important to recognize here that PAMPs and DAMPs, so whether the activating molecule is actually from a pathogen or if they are our own molecules, it doesn't really matter. The receptors can't differentiate between these things really. So they do generate the same principal downstream immune response. So there are several families of these pattern recognition receptors. And the main families are listed here. The TLRs is the biggest and most well-studied family, the toll-like receptors. Then we also have C-type lectins, and some of these are actually part of the complement system as well. And then we have NLRs, which are cytosolic receptors. So I will be talking a little bit more about the TLRs and the NLRs. And then we have other types of receptors that basically only recognize certain DNA or viral RNA. And these are receptors that have a couple of different functions. So one is to activate the transcription and production of cytokines and chemokines and other mediators that will alarm the rest of our immune system and send out an alarm and say that now there's an infection going on or some kind of cellular damage and we need help. So to recruit more immune cells, for example. And also to tell the immune cells that come in what type of infection is going on. So the cytokines that are being produced will also affect the function of the immune cells that are being recruited. These receptors can also have direct effector functions. So, for example, the scavenger receptors, which are not listed here, they are receptors that can induce phagocytosis. There are, as I said, the CLRs can lead to complement activation via the lectin pathway. And some of these pattern recognition receptors can also induce the release of specific mediators that have a direct effect on the microbe. So, for example, reactive oxygen species or nitric oxide, which can be directly toxic to the microbe that is present.
Jenny,
yeah.
Just a second. Yeah, you said that these PRRs, they have very specific targets.
Yes.
Right? And this was established during the evolution.
Yes.
The targets. Do you think it's still evolving?
Yes.
Yes. So is there any group that study this kind of evolution that you know? It's kind of interesting because since it's from evolution and it's evolving, and when we learn from immunology, we say that it is strict, so it doesn't change. But if we think about evolution, it's always like evolving and becoming better.
Yes, I think there are very few things in the human body that are not changing over time and adapting to different things, even though it's slow. There is here in Lund, there is a group actually in the Faculty for Sciences that are working with evolution in toll-like receptors, for example, and how they evolve over time. And they are catching wild mice and analyzing TLR receptor evolution in mice. So yes, there are groups that study this for sure.
Okay, thank you.
Thank you. Let's see. So toll-like receptors. So there is a handful of different toll-like receptors, and they are listed here or depicted here in this figure. So these receptors are always membrane bound, and they can be bound to the plasma membrane. So that means on the cell surface. So they are actually sitting on the outside of the cell. So they will be able to sample things that are outside of the cell. And then we have a group of toll-like receptors that sit in endosomal membranes. They are also always on the membrane, and they are always pointing inwards towards the endosome. So you can imagine something that has been taken up and internalized in an endosome, we will be able to sense through any of these intracellular endosomal toll-like receptors. They are never pointing towards the cytosol. So the toll-like receptors are strictly into surface-bound compartments. And they are localized sort of, or their localization reflect what they recognize. So if you look at these receptors here that are on the surface of cells, they recognize these here are compounds of gram-positive bacterial cell walls, flagellin, as we were talking about a little bit before, and LPS, like we also talked before. So these are all things that are specific to microbes and can be sensed in the extracellular environment. Of course, mostly this relates to bacteria. And then these receptors in here are the ones that recognize nucleic acids or different types of nucleic acids. And as you can imagine, these receptors are really good at sensing viral infections, while these are maybe more prone to sense bacterial infections. And it's actually been shown, there is a couple of individuals here in Sweden, brothers that were in their 30s and healthy, but got very sick during with the coronavirus now, just last year. And there was no real explanation for why they would become so sick. So there was a team of doctors that started looking into if they had any immune deficiencies. And it turned out that they were actually deficient for TLR7. So they had a specific mutation that made them lack this receptor, and that made them very, very sick. So it's as an interesting observation. And it's actually also important, a very important aspect of having these three receptors that can sense nucleic acids in endosomal compartments is that they're actually not very good at differentiating between human DNA and microbial DNA or RNA. So if you put these receptors on the surface of our own cells, they will actually also recognize our own DNA from cells. You have always have like a little bit of cells that die, and that will cause a reaction if you move these receptors to the surface. And there was actually a research group many years ago that did just that. They moved TLR7 and 9 to the surface of cells in mice, and those mice got horribly sick with autoimmune disease. So it's localization of toll-like receptors is very important. And when these receptors signal, they always signal as dimers. So they're present at the surface as monomers, but then when a ligand is approaching or binding, they will dimerize. And they can dimer up as heterodimers, which TLR2 is always part of a heterodimer with TLR1 and TLR6, while TLR5 and TLR4 usually act as homodimers. So TLR5 dimerizes with TLR5 and TLR4 with TLR4. It's not entirely true, it's true, but it's also, we are more and more learning that these receptors can actually dimerize in more ways than we first knew. And these different complexes also let cells sense new things that we didn't know that they could actually sense. So I think we don't have the full picture here yet. We're still learning. It's also important here that, which also makes it difficult to know. So some of these toll-like receptors, they bind directly to their target. For example, TLR5 binds to flagellin directly. TLR4, on the other hand, cannot bind to LPS directly. It needs coproteins, for example, MD2, which has to bind to TLR4 and LPS for an interaction to occur. And there's another protein called CD14, which is also required here. And as you can imagine, when this was realized, this we only know because we happen to find these other receptors, but we actually don't know. Maybe there are other accessory proteins that can interact with TLR5 that will let TLR5 act or recognize something else than flagellin. So there's definitely a lot to learn still here in this field. So what happens when a toll-like receptor recognizes its ligand? There's an enormous signaling cascade on the inside of the cell that's being activated. First, recruitment of an important adapter protein that I'm sure you've heard of, called MyD88 happens, or for the intracellular, some of the intracellular receptors, there's or for TLR3, there's another adapter called TRIF. So these proteins are being recruited to the toll-like receptor on the inside of the plasma membrane or endosomal membrane. And then that activation triggers the activation of a whole series of different kinases and ubiquitinases, which eventually lead up to the activation of different transcription factors, which are translocated into the nucleus and can then start up the production of important mediators, for example, pro-inflammatory cytokines or type 1 interferons. And in terms of toll-like receptor activation, these are the main products that we get out of toll-like receptor activation. It's the pro-inflammatory cytokines and type 1 interferons. And we have another important receptor family within the PRRs that are called the NLRs, so the node-like receptors. And this is a receptor family that is present in the cytosol of cells. So if the TLRs were mostly protecting on the membrane surfaces, the node-like receptors are surveilling the cytosol. So when something comes into the cytosol, the node-like receptors are there to recognize it and start an immune response from the cytosolic compartment. And there are a handful of different NLRs, and they all have different functions. And some of these NLRs actually have different functions in different cells. So it rapidly gets pretty complicated when you work with these receptors, because you need to know which cell type you're in and what you can expect and what you know about these NLRs within that specific cell type. And some of these NLRs can form something called an inflammasome. And an inflammasome is a very particular immune activated complex. So this is when the inflammasome activating NLRs sense their ligand. So here's an example of an inflammasome activating receptor. So this one is called NLRP3, and this is present in the cytosol of immune cells. So when this receptor senses its ligand, what happens is that it binds to a whole complex of proteins, and the first one is called it's a small adapter protein called ASC, which can then in turn bind to a protease called caspase-1, and this caspase-1 is when it's not in an inflammasome, it's in its proform and it's not active. But when this ligand is sensed, these proteins bind together, and there are a few sensor proteins and many proteins of the ASC and the caspase-1 type. And they form this wheel-like structure. And then once they have been oligomerized into this enormous complex, procaspase-1 becomes cleaved and activated into caspase-1, and caspase-1 in turn then can cleave other proteins. So in our cytosols, we have proforms, for example, of important pro-inflammatory cytokines. IL-1 beta and IL-18 are specific for this. And they are being cleaved by caspase-1 in the cytosol. And also somehow, we don't really know how yet, but they are being released from the cell. Caspase-1 activation can also cause a specific type of cell death called pyroptosis. And it's also an inflammatory type of cell death where the membrane ruptures and the cytosolic contents is released. So there are a handful of different NLRs that can create inflammasomes, and they all sense different things. So NLRP3, for example, can mostly recognize actually disturbed ion levels. So when a cell is challenged by something and you get the cell is changing from the homeostatic level in terms of ions, the NLRP3 protein is activated. We don't really know how yet, but that's an intense field of research. And then we have a couple of different receptors that are more easy to study because they bind specifically to certain bacterial things like flagellin or type 3 secretion systems. And AIM2, which is actually not an NLR, but is a cytosolic receptor that binds DNA and can also create inflammasomes. So then what different types of evasion mechanisms do bacteria have to conquer these things, to actually be able to establish infection? You can be very sure that for anything that we can come up with as humans in our immune system, there will be microbes that have ways to evade that particular aspect of our immune system. There's no way around it. So if you remember this, we were talking about what it takes to become a successful pathogen. We have these different aspects that we need to do. And now, of course, there are mechanisms that bacteria or viruses employ that are active at all of these different levels. But I will be speaking mostly now again about innate immunity. And the two main strategies, if you want to just make it very simple, that microbes use is either they interfere with immune responses or they avoid them. So obviously a good way of not triggering an immune response is to not be seen, fly under the radar. And the other way is to actually change the responses. If you have to be detected, then at least maybe you can change what happens once you've been detected. So these are the main aspects of immune evasion mechanisms. And these are just a handful of mechanisms that bacteria and viruses use, but I think most of our specific responses or mechanisms fall under one of these categories. And one of them is evasion of phagocytosis. Phagocytosis is the process of immune cells eating, for example, bacteria or viruses that we talked a little bit about during the complement system as well. So opsonized bacteria, for example, will be recognized by phagocytes like macrophages and eaten. And there are mechanisms that, for example, downregulate complement, downregulate antibody recognition, which will then make macrophages, makes it more difficult for macrophages, for example, to recognize the microbe. There are mechanisms of that entails evasion of direct recognition by specific receptors or specific antibodies. And for example, there is something called antigenic variation that most microbes use, and that I'm sure that you've heard of also now in context of COVID, where it changes and adapts a little bit all the time. So we have this Delta variant, for example, that seems to spread faster but cause not as severe disease. So that's a perfect adaptation to becoming a more successful pathogen. And within the antigenic variation can occur a little bit different by two different mechanisms. So one is antigenic shift, which is happening now in the with the Delta variants, where there's antigenic drift, which is happening with the Delta virus. So that's an accumulation of point mutations. There's a gradual change in a virulence factor, for example, within a pathogen. And then there's antigenic shift, which is happens in these influenza viruses each year, where they change an entire gene. So it's a very abrupt shift, and it's quicker, and it causes a more significant change. And then there's also, for example, this allows bacteria to avoid, or one way that bacteria use, another way of bacteria and viruses use to evade this recognition is, of course, by having their life cycle within cells that we also talked about before a bit. So once bacteria and viruses have been able to enter inside our cells, if they can create a niche in there where they can survive, then they will also avoid being recognized by, for example, Toll-like receptors or specific antibodies. There are also certain bacteria that have also actually evolved certain forms of LPS. So there's a Helicobacter, for example, or some other bacteria that express a form of LPS that allows them to evade recognition by TLR4, but the LPS is still functional. So it lets the bacteria keep their structure, but evades TLR4. So there are just a couple of bacteria actually that have managed to do this. Another way that the microbes evade mechanisms or immune responses is actually to modify the response. So if you can't evade, as I said, being recognized, you might be pretty good still if you can actually change the immune response that is being triggered by your presence. So I am going to give you just a few examples from the work that we do in the lab and the work that I've done previously as a PhD student. And I'm going to use my favorite bacterium for this. This is Streptococcus Pyogenes, or group A Streptococcus. And this is a very, very common bacterium. It causes something like 700 million superficial infections each year in the world. And most of these infections are mild infections. So tonsillitis, which is depicted here, so these strep throat, it's also called, or impetigo, which is a superficial skin infection. So these are very mild infections. They are self-limiting. If you don't treat them, they will heal by themselves, but they're most commonly treated with antibiotics. These bacteria can also, in some cases, cause severe life-threatening conditions, such as necrotizing fasciitis, which is depicted here. It's a terrible, terrible disease. We don't really know often how it happens, but when these bacteria gain access to our deeper tissues, for example, through puncture wounds, or patients that have had burn injuries are very susceptible to this, but in many cases, we actually don't know where how these conditions starts. But once they do happen, they progress very fast. So you see on this patient here that they have drawn, so this line here is the first line. This is what they could see when this patient came in. And then the infection moved to here, and then here, and here. And it's not very visible here, no, but there is actually just half an hour between these two lines here, and you can see that it's spreading here. So it's super quick. Basically, you can come in, you can recognize this in the morning and be dead by night. So this patient was fine. He had major surgery. Like all of the infected tissue was removed, and he survived and long enough to make his own website about this. So this is a bacterium that can cause extremely different infections, and that is partly due to the fact that this bacterium expresses a whole array of different virulence factors, many, many different virulence factors. And I'm talking about a few of them today, or 3 of them. One of the main virulence factors is the M protein. It's a surface protein, and you can see it in this picture here. It looks like a little fuzzy beard on these bacteria. So this is the M protein. It's a surface protein that is attached at one end and then portrays out into the environment. I will also be talking about a little bit about a protein called SLO. It's a pore-forming toxin, and it's co-toxin nadase, which is expressed together with SLO, and it's a glycohydrolase, so it cleaves NAD plus. So the M protein of group A Streptococcus is a protein that is required for the bacteria to evade phagocytosis. And this is just an example graph here. It's not an actual graph. So if you look, for example, at this is if you take some of this bacteria and you inoculate them in human blood, what will happen is that they will grow in the blood with a generation time of less than 20 minutes. So they grow extremely fast in blood, but only if they have an M protein on the surface. If we inject a bacteria where we have removed the M protein, they won't survive at all. They will be eaten actually by phagocytes in the blood. And this is almost entirely due to the ability of the M protein to recruit complement regulators and therefore block complement deposition on the bacterial surface. So what happens is that these M proteins binds to, for example, a complement regulator called C4BP. And once this has bound, it blocks complement activation, and you have less C3b on the bacterial surface, and therefore macrophages and neutrophils cannot recognize the bacteria anymore because they're not as well opsonized, and the bacteria can grow in the blood instead. What is also interesting with these proteins, so this is an example of phagocytosis evasion. This same protein is actually also used to evade recognition by antibodies. So you see here, the way these proteins are sitting on the bacterial cell walls.
Jenny, just a question.
Yeah.
So it's the M protein, it binds to C4BP and
Yes.
trap it and avoid it to activate. Is that right?
No. So see, C4BP looks like, so this is C4BP, it looks like an octopus.
Okay.
So what C4BP does is that it blocks complement activation. So if you go back here, so you remember once you have activation of complement on the bacterial surface, for example, you get C3B deposition onto the bacterial surface. What C4BP does is that it blocks the activation of the complement pathway.
Okay.
Yeah, so it's not actually interfering with the complement proteins directly. It's interfering with or recruiting this regulator. So this is what the human cells does. So on human cells, we have all of these complement regulators, and that's why we are not being hurt by complement ourselves.
Okay.
So basically, the bacteria is hijacking this regulator and pretending to be a human cell, so to speak.
Very clever.
Very clever.
Okay, thank you.
But what's also very fascinating with these proteins is that, so this is the M protein here attached to the bacterial cell wall. And at the very end terminus of these proteins, there is a region that is called a hypervariable region. And this is a region that is a target for protective antibodies. So when we get immune responses or immune to these bacteria, we will get antibodies. So if I become sick with a certain type of group A Streptococcus or Streptococcus Pyogenes, I will develop protective antibodies against that bacterium. And these antibodies will be directed against the HVR. Excuse me. So what the bacteria then is doing to evade this and to be able to keep living in the human population is that the bacteria vary this region, but only this region. So over time, this region changes so that these protective antibodies can no longer recognize this region. So with group A strep, we have about 200 to 250 different serotypes, and it's increasing all the time. And these serotypes are then different only in this HVR region, meaning that if I have, for example, there's a strain called M22, that's the serotype 22. So if I have an infection with serotype 22, I will be protected against type 22 next time I meet one of these bacteria. But I will not be protected against the M2 serotype or the M1 serotype or the M200 serotype. So as a population, the bacteria will be able to sustain themselves in the human population. But what's really cool about this is that it actually puts a dual pressure onto this small part of this bacterial protein. Because on one hand, this protein, the bacteria want to be able to keep C4BP because it protects them against phagocytosis. But they also want to, they want to keep that ability to bind C4BP. But they want to change the same region enough so that these protective antibodies are not able to recognize it. So it's quite tricky for the bacteria. So, but it does this very well. So this here is an alignment of different HVRs from different M proteins, and you can see that they vary extremely. In fact, there are only three places where they are completely conserved. And this is something that is very important, because what the bacterium has been able to figure out by evolution is that they can keep, as long as they can keep a certain structure, this structure is enough for them to bind C4BP. And these three residues here are crucial to keep that structure. But any of these other amino acids around, they can modify to basically anything. And that modification lets the bacteria evade the protective antibodies. It's amazingly clever.
And we had another question. So
Yeah.
it's asking, are these variations in serotype related to different diseases caused by group A strep?
Yes and no. So it's some of these serotypes are more or less associated with certain types of diseases. So the M1, the M3, the M89 strains, for example, are particularly associated with these nasty deep tissue lethal infections. There are also some of these serotypes that are more prone to cause these skin infections, and some are more prone to cause throat infections. But it's not clean cut, so you can always basically find all types of serotypes in any disease. Mm-hmm. But there are certain M types that are more associated with some than others.
Okay. Mm-hmm. Thank you.
Thank you. So that was the M proteins and the evasion of phagocytosis and evasion of recognition by antibodies. So group A Streptococcus or Strep Pyogenes also have the ability to modify cellular responses. So this is also something that we've been working on in the lab. And group A also, Strep also produces these two secreted toxins. So SLO and NADase are two toxins that are secreted and released from the bacterium. And SLO is a toxin that can bind to host cells and form a pore. And NADase is a protein that can enter host cells and cleave NAD+. Or these are the original things that we knew about these proteins. So what we've been working with is it was found, not by us, but by other researchers many years ago, that SLO, or streptolysin O, as it's also called, actually activates the inflammasome. So activates the inflammasome that we just talked about, which then generates a release of IL-1 beta. And IL-1 beta is required for our ability to protect ourselves against these severe infections with Strep Pyogenes. So there are patients, for example, that are giving treatments for certain autoimmune diseases, such as certain joint diseases like rheumatoid arthritis, for example. They are given medications that will block IL-1 beta. And these patients, we've realized that they become very sensitive to these deep tissue S. pyogenes infections, which is very unlucky. So they have a 500-fold increased risk of developing severe infectious disease or severe group A strep disease. So what people then have found is that SLO, as I said, activates the inflammasome, and this you can see here. So this is our own data in mouse macrophages from different mouse models, and the B6 is the wild-type mice, and these you can ignore. These are just different inflammasome proteins. And so wild-type bacterium, when you use them to infect macrophages, you will generate IL-1 beta production. And this production is completely dependent on the presence of SLO. So this is a bacterium that lacks SLO expression. So you won't get any IL-1 beta. Now, what we also found here is that when you're infecting macrophages with cells that, with bacteria that lack NADase, this NAD glycohydrolase, we surprisingly found that these macrophages can make a lot more IL-1 beta. So the way we interpret this is that when you have a functional NADase on your bacterium, this actually limits the amount of IL-1 beta that is being released. And this is a little bit complex. So on one hand, these bacteria activates the inflammasome to generate IL-1 beta release. On the other hand, NADase is also limiting the amount of IL-1 beta that's being released. So the way that we see this or look upon this is a little bit of IL-1 beta is probably good for the bacterium. So opens up the tissue a bit. So IL-1 beta is a protein that's able to create, for example, permeability in the vasculature. So it makes it easier for the bacterium to actually enter the tissues. But too much is not good, because if you have too much IL-1 beta release, then that's bad for the bacterium. So, but just a little bit is enough. So we see this as a clever way of the bacterium to balance the amount of IL-1 beta that's being released for its own benefit. We're not entirely sure how this happens, but what we do know is that somehow Strep pyogenes is interfering with a second surface receptor called P2X7. And this is actually an ATP receptor that is then recognizing high amounts of ATP in the extracellular environment. So if you see here, this here is infection of macrophages from wild type or P2X7 deficient cells, mice. And then the wild type bacteria causes IL-1 release as normal release in both cell types. But if we use bacteria that lack NADase, this NAD glycohydrolase, we get the increased release of IL-1 beta from the wild type cells, but not from the P2X7 deficient cells. And our interpretation of this is then, of course, that P2X7 is actually required somehow for this additional release of IL-1. We don't know how, but it seems to have to do somehow with interfering with the release pathway. So there are multiple ways that IL-1 beta can be secreted from cells. And this seems to have to do somehow with interfering with these release pathways. So another way that Strep pyogenes is interfering with IL-1 beta in particular, so as you can imagine, this is a very important target for the bacteria to hit, because it has multiple pathways to direct evasion of IL-1 beta in particular. So another thing that we've found in the lab is that when we infect macrophages with Strep pyogenes, wild-type Strep pyogenes, as you see here, or bacteria that lack streptolysine O or SLO, the wild-type bacteria induces a significant amount of ubiquitination on IL-1 beta, pro-IL-1 beta, while the SLO mutant does not induce any ubiquitination of pro-IL-1 beta. So IL-1 beta is a cytokine that is present in the cytosol as a pro-protein, and that protein is not active. It has to be cleaved by the inflammasomes to become active. And this is then ubiquitination of the pro form of IL-1 beta. And what this leads to, interestingly, if we infect macrophages and we leave them over time, what you see here is the bars representing infections with the wild-type bacteria is the amount of IL-1 beta is continuously decreasing. So ubiquitination, of course, is an important molecule in our systems that clean up in our cytosol. So dying organelles, for example, are ubiquitinated and sent to the proteasome for destruction. So we think this is what is happening here as well, that the cells that are being infected with the wild-type bacteria, somehow we have a targeted ubiquitination of IL-1 beta, and we think that this is something probably that the bacterium likes, although we're not sure at this point. But the pro-IL-1 beta is ubiquitinated and then targeted for destruction. And that, of course, limits the amount of IL-1 beta that the cell can release into the extracellular environment and send off the alarm to neighboring immune cells and say that, hey, there's an infection here. Come help me.
Jenny,
yeah.
Just another question. So the bacteria, it releases the SLO, streptolysine O, once it's inside the macrophage?
Yes.
So, okay, so it must be like an Intracellular pathogen.
Sorry. No. So both. So it releases SLO at all times.
Mm-hmm.
So it would be in the extracellular environment, but it's also released. So if Streptococcus pyogenes is actually eaten by macrophages, it will continue to be released. So it's actually active also on, it can rupture the phagosomal membrane.
Yeah, but for it to interact with IL-1 beta, it should be inside the cell.
So SLO in itself is not interacting with pro-IL-1 beta. It's the presence of SLO is causing ubiquitination of pro-IL-1 beta, but SLO as such is not actually binding to IL-1.
Okay. Thank you.
Thank you. Okay, so that was actually my last the last thing that I want to say. I'm realizing that I've been talking for a long time, much longer than I expected, and I apologize for that. But to sum up quickly, the take-home message from today, I would like to be for you to remember that successful pathogens has to be able to spread within the human population, which means that they most commonly cause pretty moderate disease. And also that most successful pathogens employ several different types of evasion strategies that are directed at different aspects of the immune response mounted by the infected host. And in specific terms for innate immune responses, these are responses that are very quick, so they are part of our most acute response, but they are non-adapting, so they're not very clever, and they don't get any better with time. And we have both humoral and cell-mediated parts of innate immunity. So, for example, the complement system in our fluids, the blood, and pathogen recognition receptors on our innate immune cells, and they have different functions, and they're covering different localizations of the cell. So thank you very much for listening, and thank you for your questions. And if you have more questions, then I'm happy to take those now.
Okay, thank you very much, Annie. It was a really nice presentation. I think it helped a lot for our students. So the post-graduation students, they just had a few weeks ago class regarding this innate immune response, and I think it was really good for them to take like a big picture of it. Yep, it was really nice.
I'm glad you enjoyed it, and thank you very much for all the questions. If you have any other questions that you don't come up with now, but you want to ask me or talk to me about something else at some point, please don't hesitate to contact me. I'm always accessible via email.
Oh, thank you very much.
Should I go out of my presentation mode now?
Please, if you could.
Yes, please.
So thank you so much again for joining us today. We get great comments on your talk. So it was an excellent talk. I think it was very clear to the students in a very nice level for them. So thank you very much. And of course, thank you.
Thank you very much.
So I'm not sure. I think, does anybody else have any questions for Dr. Jenny while she's still here? Because she said she has another appointment. So yeah, the students are just making comments about how nice it was. Thank you very much.
Good. And thank you again, everybody, for listening. And thank you, Tiago and Michelle, for inviting me. It really was a pleasure.
Thank you.
Good luck with the rest of your series.
Thank you very much, and good luck with your research.
Oh, thank you very much, and to you.
Take care. Bye. Então, pessoal, nós queríamos chamar um breve intervalo de 5 minutos. Pedimos que vocês aguardem aqui no canal mesmo. E nós vamos entrar agora com a próxima apresentadora. Em 5 minutos nós retomaremos com a apresentação dela. Muito obrigada pela participação até agora. Até logo. Hi, Serena. How are you?
Hey, good. Thank you. And you?
Very good. Thank you. Very well. Thank you so much for joining us today. I'll just go through the presentation very fast with you so that we have all the details. So if you go on the share,
Yeah.
then you have share screen. It's the best way to present.
Okay.
And then you can share your whole screen. And when you start, you will leave this room and you move to your presentation. So you will not be able to see us, but you can use your pointer. It's working well. Okay?
So
and then when you finish, I'm sorry?
No, because I will keep the presentation. Okay.
And Marcela is going to share your presentation with the audience so you can present freely. And then when you finish, we can ask the questions. I think it would be better instead of interrupting you. Is that okay?
Yeah. Yes, it's okay for me. Yeah.
So I'll just make a quick introduction about you.
Yeah.
Before we start, and then I'll let you go on with the presentation. Thank you so much again. It's great to see you.
It's nice to thank you for the invitation. Shall I share directly now the screen? Or
you can do that, yes.
Yeah.
And then I'll present you. So I'll do a little Portuguese with English mix, if that's okay. And mille grazie. Então, pessoal, bom dia a todos novamente. Nós estamos retornando agora com a apresentação da doutora Serena Bettoni. Vou fazer uma breve introdução, então, da doutora Serena em inglês, né? E aí ela vai continuar com a explicação aí do trabalho dela. So, hi everybody. Welcome to the second talk of our day, our International Microbiology Workshop. We have here now Dr. Serena Bettoni. Serena is from Italy, and she's actually now doing her postdoc studies at Dr. Anna Blom's lab, where she's investigating the interactions of the complement system with pathogenic bacteria. And she's going to talk today about Neisseria meningitidis. So thank you so much, Serena, for having you today. I had the pleasure of working with Dr. Serena when I was in Sweden, and I'm sure you will enjoy her great work. So it's an honor for us to have you here today, and you can start whenever you feel like it. Thank you very much.
Okay, so thank you, Michelle, for the invitation. It was nice. And can you see my screen now? Yes?
Not yet. Marcela, você pode, por favor, compartilhar a tela da doutora Serena? As soon as I see your screen. Oh, I think it's not showing yet. So if you can press share and then share screen.
Yeah, I did it.
Could you try again?
Yeah. I think.
Let's see if it shows now. It's not showing yet. Let me see. So the share button, I think it's on the...
Yeah, I'm clicking on it and then share screen, and then I click on Windows and Microsoft PowerPoint, and then share. And
could you try to leave it open, the PowerPoint, and then go back here?
It's open, actually.
Yeah, this thing's usually, it's part of the whole online world, I guess. There's two types of share. There is one that says screen and another one that says video archive or something.
Yeah, I'm using the share screen, actually. Is that okay?
Okay, so Marcel is telling me that you should, it's going to show you, like within this tool, the screen presentation, and then you click on it, and then you click on share. So
I don't understand why it's not working then. I'm clicking on it.
If you would prefer, you can try and send me by email your presentation. That's also okay. The only thing will be the pointer. I can share it for you, and I'll go through the slides for you.
I don't know why.
So what I cannot see anything. So what Marcel is telling me is that you have to click on first. Let me see.
Share.
Yeah.
And then
So,
share screen.
yes, so it's share within the StreamYard, and then you click on share screen. And if your PowerPoint is open, it should be showing by now.
It's not happening.
When you press share screen, does it appear the PowerPoint presentation on a dialog box?
Yeah.
And when you click on it, it's not showing?
No.
Let's see what we can do. Just a minute. So, yeah, I think it would be better if you would leave us and try to log in again, and then see how, yeah, thank you. Então, pessoal, peço que aguardem um minutinho até ela conseguir conectar a apresentação. Estão gostando? Muito boa a apresentação da doutora Jenny, né?
Bom dia a todos. Vou aproveitar esse breve intervalo para agradecer a professora Michele, ao professor Tiago, por toda a organização do evento, a equipe da USF que está dando suporte também. É uma oportunidade excelente não só para atualizar os conceitos, né, mas para ouvir um pouco da ciência em inglês, e isso é tão importante para todos vocês. Então, espero de verdade que vocês possam aproveitar, mesmo que não seja exatamente o tema da sua área de estudo, o máximo possível, e participar com questões simples, questões em português que sejam, mas que demonstram aí o interesse de vocês em entender essa mensagem. Agora, a doutora Serena já retornou, né, e a gente vai tentar compartilhar novamente a tela.
Isso, obrigada, Andrea. So, Serena, could you try again?
Yeah. Share. Do you see?
No, still nothing.
I don't know.
Yeah, it should be appearing. So when you click on share screen, what does it show? Can you go through it with me so we can just...
So share screen,
Yes.
sharing tips, and then share screen, and then 3 options: entire screen, Windows, or Chrome tab. And then
Okay, which one are you choosing?
entire screen.
Okay. Do you want to choose the second option and see if that helps?
Yeah. Can you see? No.
No, not yet. So... I'm not sure why it's not showing. So what we can try and do, Serena, so we don't you can just send me your presentation and I can try to share it from here, that you will not be able to... Okay, we can try another thing. While you send me the presentation,
Yeah.
I will try to open a Meet with you, and we can try to share our screen. Just 1 minute. So let me see. Serena, do you see this private chat on the right? I just shared with you a Meet screen. If you could click on that.
Yeah.
And share your presentation there. Have you worked with Meet before? Google Meet. It's like this arrow pointing up. So you click on the arrow pointing up.
Okay. Impossibile condividere to see. I cannot.
What we can do, I'll just check if you send me your presentation.
Yeah.
And I can try to share it from here.
Can you see it now? In Meet?
Yes, on the Meet screen we can see it.
Okay, so I will try again to the other side.
Let me see.
Can you see now?
I can see it on the Meet, Serena, but not on the StreamYard. So now I'm going to connect them both. Just a minute.
I think it's sharing my screen now.
Okay. So is it working on StreamYard?
I think it's working now.
Okay, so I'll just remove my.
Yeah.
Okay, so yes, looks like it's working. So please, Serena, whenever you feel like it.
Okay, so yeah, so if you can see my screen, I can start.
Yes, we can see it. Thank you.
Yeah, that's great. Thank you. Yeah, so I'm Serena Bettoni, and I'm a postdoc researcher in Lund University in Sweden, working with Professor Anna Blom, as you told already. And so my talk will be a bit more into data and results in practice in the laboratory techniques in comparison to Jenny talk. And I want to show you about results that we have regarding the C4BP IgM fusion protein used for treatment against infection, especially for Neisseria gonorrhea in this case. So Neisseria gonorrhea is a pathogen, a bacteria, and is a diplococci. That means that is usually seen at a microscope, in the microscope, as two joined cells with the adjacent side flattened like this. So looks like a coffee bean shape, and is considered as fastidious pathogen because require specific enriched media, especially for CO2 atmosphere and 35 to 37 degrees. And this enriched media, usually in the lab, is chocolate blood agar. I show here a plate that is like an example. That is a non-selective enriched growth media containing red blood cells that have been lysed slowly at 80 degrees. So the cells release iron, that is an essential component for bacterial growth. And so Neisseria gonorrhea is a gram-negative pathogen, exclusively pathogen, and can cause an infection that is a sexual transmitted infection called gonorrhea, so same. And so usually the infection is asymptomatic and can solve per se alone during in the body, but if persists and is not treated, then can cause some bad consequences, especially in women, for example, infertility or neonatal blindness here. Okay, regarding the treatment of this disease, so nowadays there is no vaccine. And why? Because the immunity is strain specific, and the the antigens used for creating a vaccine, they are highly variable. So, for example, porins, that is the major protein on the membrane of bacteria, on gonorrhea. And also, gonorrhea doesn't not express capsule, which is the, let's say, a wall that surrounds the bacteria and protect the bacteria, and which was actually a successful antigen for the development of the vaccine for Neisseria meningitidis. So nowadays, since there are many failures for the clinical trials for the vaccine anti-gonorrhea, companies are like discouraged to give money to this to new development of new vaccine. And the current treatment is based on the use of antibiotics. In 2010, the World Health Organization gives some recommendation to use the third generation cephalosporins and in combination with azithromycin. But already after 2 years, they remove cefixime, that was one of the cephalosporin. They remove cefixime from this list because of the resistance that bacteria developed. And nowadays, there is a new guideline giving ceftriaxone as a monotherapy for the first line of antibiotic treatment for this infection. But this will not last long as well, because the bacteria, so Neisseria gonorrhea, is becoming resistant to antibiotics very fast. And if we see this graph that is coming from a publication here in 2014, that shows that since the first time that gonorrhea was treated, so we are talking about 30s, almost all the classes of antibiotics, here there are some, have been used, and all of them have been also discarded because bacteria, gonorrhea, got resistance. So now they cannot be treated anymore with these antibiotics. So a new way of treating gonorrhea infection is needed and should be suggested. Entering a bit in my results, so the topic will be related to the innate immunity and humoral immunity and the complement system. And Jenny Persson before explained very nicely to you what was the complement cascade, what is the complement cascade. So just a brief reminder, complement cascade is a huge cascade of proteins happening in the human body and can be activated by 3 pathways according to the stimuli that is coming from the outside. And these pathways are lectin pathway, alternative pathway, and classical pathway. These 3 pathways converge in the activation of the central enzyme, that's called C3 convertase here, which then create another important enzyme called C5 convertase, which in turn leads to the formation of the membrane attack complex, also called MAC. And MAC is this like big protein complex that create pores, as we can see here, like a barrel. And this insert in the membrane of cells and create and allow liquid and salt to pass. So the osmotic balance is disrupted, so the cell die because of lysis. So this is actually what happens for gram-negative bacteria. So the complement is activated immediately when the infection is starting in the body, and then MAC is formed and then sits on the bacterial membrane, gram-negative bacterial membrane, and then bacteria burst and die, and they are eliminated. So this is usually what should happen, but various bacteria, and gonorrhea is one of those, evade this cascade. How? Basically recruiting C4BP. Jenny also mentioned before. So C4BP is an important soluble complement inhibitor. It's a big protein that stops the cascade at this level, so at C3 level. So prevents the formation of C3 convertase. So bacteria that has C4BP on the surface bound to porins, which are the most abundant protein on the surface of these bacteria. So the complement is blocked, is inhibited. So the bacteria can survive and promote infection. So then what we did some years ago was to check the prevalence of the binding of C4BP by various clinical isolates from Neisseria gonorrhea. So among a large collection of isolates isolated from patients with sexual transmitted infection that we in Sweden in the last 10 years, we got 190 strains. And then we checked for the C4BP binding. And what we saw was that almost 90% of the porin 1A, so the serotype porin 1A, binds to C4BP, and 20% of the strains for serotype porin 1B binds to C4BP. And look into the anatomical site of isolation of these isolates, we couldn't see a clear correlation between C4BP binding and the site of isolation of the bacteria. But what was clear was that when bacteria are binding C4BP, so let's say here in this area, they also survive within serum. We use serum, so here the acronym NHS is normal human serum. That is the source of complement, the complement cascade that I was mentioning to you before. So usually bacteria are killed in serum, but gonorrhea survive. If binds C4BP, then survive against serum, as we can see here. There was some strains actually here that they were not binding C4BP, but still able to survive. And then we found out that these strains, they were binding another complement inhibitor called factor H, FH, written here, that is also blocking the cascade at C3 level. So in the same mechanism. So what we thought then was that maybe to have a new way of treatment of bacteria, we can use this evasion strategy that bacteria develop to survive. We can use in our manner, so in our convenience. So what we did was to create a fusion protein. Fusion means that two proteins fuse together, and we choose the C4BP because it binds to bacteria, and especially we use these two in yellow, these two domains that are able to bind the porins of gonorrhea, and we fuse with the constant portion of the immunoglobulin IgM, here in red. So the construct that is originated is actually becoming hexameric, as we can see here. So 6 arms of the same molecule. And this is also convenient because can compete with the heptameric, so this octopus structure of C4BP. So what happens is that this molecule here in red, called C4BP IgM fusion protein, binds to the bacteria and remove C4BP from the surface, because compete out C4BP. And then through the IgM portions here in red, promote complement activation through the classical pathway, and that's why here I wrote C1. So the classical pathway activates the complement cascade, so a cascade of deposition of complement until MAC generation, so the pores, MAC pores generation. And therefore, the bacteria die by lysis. So then, in collaboration with the University in Massachusetts, that we showed that this C4BP IgM promotes killing of gonococci. So we use a panel of some 20 isolates, and as we can see here, so they are all resistant to killing. So they survive completely, as you can see, survival CFU per ml in serum. So the black bars. So they all survive because they are able to bind C4BP. But when we added C4BP IgM fusion protein in red, as we can see here, the majority of them has impaired survival. So they die, as we can see here. And this is a reference laboratory strain. And moreover, then we tested this fusion protein in a mouse model infected with gonococci, so with some strains of gonorrhea, MS11 or FA1090, and both able to bind C4BP. And as you can see here, for the red lines that indicate that infection was eliminated already at day 4 for both mouse infected with bacteria. And usually the infection was released at 8 days.
Serena,
yes.
I have a question regarding the last graph. Could you please go back? Is it okay if I ask a question now? Or do you prefer in the end? That's okay. So about that sample number 9, could that...
Yeah.
So number 9 was surviving. Is that because it has the FH binding capacity, or did you check on that?
Yeah, so for some of them, we saw that they are actually binding factor H, but for others, we don't know. For example, the 22, we don't know why.
I see. And is there a strain that can bind both, like FH and C4BP?
Yeah, yes, they can both bind C4BP and factor H, but importantly, in vivo, gonorrhea can become can acquire sialic acid from the human cells, so and then can be covered by sialic acid, and then like to mimic that they are host cells, and the sialic acid then can bind factor H. So in vivo, all bacteria can bind factor H at the end.
Oh. Okay, thank you.
Yeah, so next idea that from here, so since C4BP IgM destabilize the membrane integrity of bacteria, then we wondered whether C4BP IgM could be used as adjuvant for antibiotics, since the antibiotics are the first line of treatment of this infection. And we know the antibiotics are internalized so they are going to the target in bacteria by direct diffusion through the membrane, or by passing through the porins, that are these abundant membrane proteins of bacteria that are used by bacteria to sampling and tasting the environment. So our hypothesis then was that since C4BP IgM here in red creates deposition, induced deposition on bacteria and then create MAC pores, membrane will be damaged. So the antibiotics influx inside bacteria can be favored and then promote killing. So then the next essay were done to prove this hypothesis. And first of all, what we did was to take this strain called Neisseria gonorrhea F8N90. That is a laboratory strain that worldwide is used for this kind of assays, and was incubated with serum, so normal human serum, that is the source of complement proteins, together with the fusion protein C4BP IgM here, and then addition of antibiotics. Then bacteria with this mix was incubated for 1-2 hours in 37, and then the live bacteria were counted, so a colony forming unit for ml, so the counting of a live bacteria after 1 day growing on chocolate blood agar was estimated, and then here are the results. So first of all, we use spectinomycin as antibiotic, and we use concentration of serum and C4BP IgM that alone didn't affect so much the surviving of bacteria. But when C4BP IgM was added together with serum and spectinomycin, here we see the red bar, survival of bacteria was strongly affected, significantly affected, suggesting that these two components, so complement, C4BP IgM, and antibiotic, work together to promote killing of bacteria. And this was happening at 1 hour. And this effect was much more pronounced when we look at 2 hours of incubation, when I could barely count any colony on the plate, as you can see here. And the same was happening with other antibiotics, azithromycin, ciprofloxacin, cefixime, and ceftriaxone. If you recognize, some of them are actually the one that the World Health Organization suggested for the treatment of the infection in human. And so then to understand the mechanism, how that was happening, so the hypothesis was that the membrane was damaged by MAC, so to prove that MAC deposition creates damage on the membrane of bacteria, we then decided to use Cytox green, which is a dye that binds to DNA of bacteria only if the membrane is ruptured. So there is a damage on the membrane. So it then can go inside the bacteria. And then after this damage, all the bacteria become fluorescently labeled, let's say, and they can be quantified by FACS, by flow cytometry. So for this assay, so first of all, we verified that the addition of C4BP IgM induced the complement deposition. So as we can see here, so deposition of C3 and deposition of MAC. Oops. And this was actually dose dependent, as we expected, and that is significantly higher in comparison to without the protein. And then when we look at the membrane rupture, so this fluorescent dye, if this fluorescent dye was going inside during MAC deposition, we actually saw that increasing concentration of the protein, C4BP IgM here, that we saw that induced MAC, induce also uptake of this fluorescent dye, suggesting that MAC creates damage of the membrane, therefore Cytox green is going inside. Okay, then we wanted to see if also the antibiotics could go inside the cell through this damage, so through the pores or other damage on the membrane. And for this reason, we took advantage of a collaboration with a group in Australia that developed this nice tool. So they linked antibiotics in this case ciprofloxacin, with a fluorescent molecule called NBD, that is like a very small molecule, that is actually here, that is smaller than the normal big fluorophores, rhodamine or Oregon green, that are usually used for labeling proteins. So here at the end we have a small molecule that is actually colored green, emit fluorescent, and so that is linked to the antibiotic. So this is actually a fluorescent antibiotic that could be used for verifying that is going inside the bacteria. And then what we did was to treat, again, the same strain, FA1090, with this fluorescent labeled antibiotic, together with a staining of the membrane, just to visualize the bacteria. And then we went to the microscope, and we saw that, as you can see here in green, the antibiotic was inside the cell, and the cell was visualized by the red staining of the membrane, as you can see here. So then we wanted to see the effect of complement, and in particular C4BP IgM, on this internalization of the antibiotic. So again, we used the same strain, FA1090. We added serum, so complement proteins, and C4BP IgM fusion protein, together with the fluorescent antibiotics. So monitoring at 15, 30, and 60 minutes in flow cytometry here, or checking the survival, checking the survival. And what we saw was that, so on the left side, we have the results for the antibiotic that I already described you. And then we use also another antibiotic that is actually a derivative of the same antibiotic that is actually used in clinic, so azithromycin, labeled again with NBD, so it's color in green. So when, so what we saw was that here in the black bars, so the antibiotic was going inside the cells, as you see the increase over time, and the addition of C4BP IgM promotes and accelerates this uptake. Here you can see the red bars. And if we look at the survival of bacteria together done in parallel in the same experiment, we could see that when the C4BP IgM was present together with serum and antibiotic, bacteria were less live, so they were killed, suggesting that the antibiotic was going inside the cell when the complement was active, and then once inside, they were killing bacteria faster because the uptake was faster. And that was happening for both antibiotic tested. So then another way to test if an antibiotic is doing and going inside the cells was actually to check if the antibiotic is reaching the target. So every antibiotic in general has a target, so a final protein to go and inhibit. And in the case of azithromycin, is the protein synthesis. So for this purpose, we use this test that I'm going to explain to you now. So again, the same bacteria, FA1090, incubated with serum and C4BP IgM and antibiotics, and a modified amino acid that has a alkene group called APG alkene here. That is the first step. During the growth, bacteria internalize and incorporate this group in the newly formed proteins, creating these alkene group proteins that then are labeled with a second step called Clique Chemistry or coupling chemistry, and that's why this technology is called Clique Chemistry. So this azide that is linked to a fluorescent dye is going to chemically bind to this alkene group, creating a fluorescently labeled protein. And what is happening at the end is that all the bacteria are becoming green. All the proteins inside the bacteria are completely labeled with this fluorescent dye, as you can see here in the confocal microscopy. And DAPI is just to check the DNA, just to see and localize the bacteria in blue. And so regarding the effect of C4b PIGM here, then what we did was to check the quantity of the protein fluorescently labeled. Here you see Lexafluor 488 protein, so the fluorescently labeled protein. And so addition of serum alone didn't affect anything, any protein content. As you can see here, that all the proteins are still there and the bacteria are still alive. When azithromycin was added in high quantity, so 0.25 microgram per ml, all the bacteria were dead because all the proteins was not synthesized. So as you can see here, the lane is empty. But when we use less quantity of azithromycin, so the sublethal concentration of the antibiotic, only partially there is only partial inhibition of the protein synthesis, as we can see here. But when we added serum plus C4b PIGM in red, again we have a full inhibition, almost big inhibition of protein synthesis, suggesting that when we add complement and the fusion protein, the antibiotic is more active in blocking the protein synthesis. And this is significantly reductive. Okay, so as I told you before, gonorrhea has developed during years a lot of resistance mechanism that can be divided in 4 categories. So these are the major determinants. So how a bacteria can survive against antibiotics. So first of all, they inactivate antibiotic by enzymatic cleavage. So they are cleaving the molecule and the antibiotic is not active anymore. Second could be genetic or an enzymatic alteration of an antibiotic target. So the antibiotic cannot recognize the molecule that is targeting, so is completely ineffective. Or reduce permeability of the cell barrier, so with the thick cell wall, or for example, the production of the capsule that protect the bacteria to the molecule from the outside, or as fourth, enhanced active removal. And regarding this group, gonorrhea is known to have 5 groups of efflux pumps. What are efflux pumps? They are like literally pumps that are pushing out from the cell the molecules that are not needed for the cells. So, and among those we have antimicrobials. So one of these group of efflux pump is called MTR, that is here in blue, and this one is often overexpressed. So is functioning very much in comparison to the normal bacteria, and when it's overexpressed by mutation, it generates antibiotic resistance because the gonorrhea is continuing pushing out the antibiotic, and antibiotic cannot kill the cells from the inside. And usually this is happening for the azithromycin resistance. And azithromycin is one of the antibiotics that was suggested to be used for gonorrhea infection. So then we decided, okay, so far we did all this experiment with a laboratory strain, but we know that in real life there are bacteria that are resistant, and they are resistant to azithromycin, and they have this overexpression of the efflux pump. So we look for some strain, and we found, so our collaborator has a huge amount of strains in Orebro, and he shared with us these 2 strains, FL29 and SA94, that they were two bacteria strain isolated from patients in Brazil, actually, affected with sexual transmitted infection. And these bacteria were resistant to azithromycin, and they have this mutation in efflux pump. So then we decided, okay, we will test the same thing that we did before with these two strains. And first of all, we verify that they bind C4BP, and actually here we see the binding, and C4BP IgM, the fusion protein that we created. And then we check also the survival of this bacteria, and so increasing concentration of C4BP IgM added in the serum, so in the complement protein source, induce killing of bacteria, because the surviving here is reducing in dose-dependent manner. So more protein was added, more killing we obtain. And this was 1 hour, and this was after 2 hours. So then we use this quantity of the protein that is not inducing killing completely, but is like a sublethal concentration that then was tested together with antibiotics. So we know that this strain is resistant to azithromycin, and we can see here in the black dots that remain at the same level. So we are looking at the surviving of bacteria. So quantity of bacteria here from the beginning remains the same, even though we increase the concentration of azithromycin, as you can see here, up to 16,000 nanogram per ml. Nothing can touch them. Then when we added serum, so normal human serum here in gray, survival was affected because the curve goes down. So quantity of bacteria recovered after 1 hour was less than the input bacteria. But when we added our fusion protein, so C4BP IgM, the quantity of bacteria reduced very fast and was completely disappeared completely after 2 hours. So all bacteria were killed at the maximum quantity of azithromycin. So what we conclude for this experiment was that serum and fusion protein, together with antibiotic, they cooperate to kill bacteria. So there was a cooperation, synergy. And the same thing that we saw before with the laboratory strain. And then to verify that was mediated by complement, so complement activation, we use a specific inhibitor of complement that is called OMC. This complement inhibitor blocks the cascade, so the complement cascade that we saw before, at the last part, so at MAC level, so when the pores are forming. So we block there, and we saw that, so the mixture of protein, C4BP, serum, and antibiotic killed the bacteria, as you can see here for the red bars. But when we added the inhibitor of the complement, the survival was rescue, as you can see here. And also when we heat inactivated serum, that completely consume all the complement, as we can see here. Okay, so then we verify that was complement activation. Okay.
Serena?
Oh,
Uh,
I'm logged out. No?
no, you're still here with us. I think just the presentation. Yes, just put it on. Yes, perfect.
Yes. Okay. Yeah, sorry for that. Yeah, so the last thing that we did was that we wanted to see if the mix of serum plus C4BP IgM could promote the influx of the antibiotic, fluorescent antibiotic. So before I show you that group in Australia create for us this antibiotic that are fluorescently labeled, called ROXY-NBD, and this is actually a derivate from azithromycin, that is one of the antibiotics used against gonococcal infection. And so we added all together and monitored if this fluorescent antibiotic was going inside in bacteria by flow cytometry or checking the survival on chocolate blood agar. And so this bacteria, as I mentioned, is resistant to antibiotic because it has this efflux pump. And indeed, as we look at this bar, this antibiotic is not going inside. I mean, it's going inside, but it's pumped out immediately, so it cannot be accumulated. That's why the bacteria is resistant. Then we tested this CCCP, that is an inhibitor of the efflux pump. And when we block the efflux pumps, antibiotic, the fluorescent antibiotic, accumulate inside the cell. As we can see here, increase very much up to 120 minutes. And the addition of serum and C4BP IgM in red also promotes accumulation of the fluorescent antibiotic inside without adding the efflux pump inhibitor. So suggesting that the complement together with the fusion protein promote damage on the membrane and then help the antibiotic to go inside and kill the bacteria. As we can see here, the red bar is going down, so the survival of bacteria is very affected, significantly affected over time. And so, in conclusion, then we demonstrate that there is a synergistic effect between C4BP IgM, that is a new fusion protein that we created, with the antibiotic in killing gonococci. And the synergy was mediated by complement because C4BP IgM creates pores, MAC pores, through the complement that can facilitate antibiotics to go inside and to explicate their activity. So let's say that C4BP IgM could be used as an adjuvant for the antibiotic treatment against gonorrhea. And that could represent a new way of treating bacteria. This is just an example for gonorrhea bacteria, but if we think, like Jenny Person mentioned, group A streptococci that are also binding C4BP, but other bacteria are binding C4BP, and maybe can bind C4BP IgM as well. So this could be also translated to other bacterial infection. And this is a new method, a new way to treat bacterial infection in a way, without antibiotic. And the fun fact is that bacteria can develop antibiotic resistance, but in such a case for C4BP IgM, they cannot, because they bind C4BP to survive. So they will not lose the ability to bind C4BP. That's why they will not lose the ability to bind C4BP IgM. So I conclude here, and this is the group where I work, and in Sweden. These are foundings, and I want to thank you all for listening. And if you have any question or comments, please ask. I'm happy to take. And I just want to add something that the group now is looking for 2 postdoctoral researcher, one in cancer and one in infection. So please spread the word.
Thank you so much, Serena. And what a nice invitation in the end, guys. Quem aí se interessou em ir para a Suécia, eu mesma já estive lá, muito legal. Então, fica aí o convite para quem estiver interessado aí no pós-doutorado, né? So, Serena, thank you so much for a great talk. It was great to finally hear you speak about your work. Beautiful work. Congratulations. And I was thinking that you made a Trojan horse, right? It's like no matter what the bacteria does, it's like a Trojan horse, you know, that horse that.
Yeah.
So, because it's such a non-specific way of reversing antibiotic resistance, you can add it to reverse pretty much any kind of antibiotic resistance that the bacteria has, right? Because they're not acting at the mechanism that the bacteria uses to defend itself from the antibiotic. You're just creating an additional pore to make the bacteria susceptible. So it's
exactly
very neat.
It's a very clever strategy, exactly. So this can also apply with other fusion protein. This is one example, but if we link other protein that bind to bacteria and then induce the complement there, creating a damage in the membrane, then the membrane is already feeling not very good. So then creates these pores and little damage. So antibiotic can go inside quickly, faster, and easier. So it's like a simultaneous cooperation. And actually now the new treatment are using various molecules together. It's like a combination of various agents. So maybe this is the key for the future, acting on various prospects.
Yeah, it looks amazing. Ana Maria had a question. She asked, she's actually interested in knowing how these molecules were made. So how do you construct, is it a genetic fusion, or do you make a chemical reaction to produce this C4BP with the antibody? How do you make it?
Yeah, so we took the gene portion of the C4BP that we know, that binds bacteria, and we fuse in the gene the portion of encoding for the constant portion of IgM. So we create this DNA construct inside a plasmid, and then we made express the cells so we can create the protein in the correct conformation that was like secreted, and then we purify the protein by chromatography, so specific chromatography. Then we check the quality of the protein, so by Western blot, or then we also check in electron microscopy to see the shape of this protein that was actually hexameric, similar to the IgM structure that has 6 arms, and then we use that protein on bacteria.
Great. So it's a genetic fusion then that you add to. And what is the host organism? Is it human cells? Is it cell culture?
We express in human cells, that in CHO cells, so Chinese hamster cells.
Okay.
That express a lot,
Oh, so it's good. Yeah, that was my next question. So how expensive would be to use this is in a clinical setting, I would say. Do you have any ideas of?
So the expense, so I have no clue how much will be in like company point of view, but the tricky part in this thing, and especially for this protein, is that the purification for the IgM part, because it's very delicate, could be affected by pH and could be affected by temperature. So so the process, the development of this protein should be very carefully considered. But not so much.
Great. And have you patented the construct yet? So how is it in terms of selling it or?
So our collaborator in America was going to, so is in the process of patenting also another fusion protein like this. So far it's not patented yet.
Okay, so it's in the development stage yet.
Something.
But you have amazing results, so I'm sure it will be a success. Does anybody else have any questions to Dr. Serena? Gostaram, pessoal? Se alguém quiser perguntar em português, pode falar também, deixar aí no chat, a gente traduz. So Serena, thank you so much one more time. It was great having you here today with us. It was an honor hearing you talk. And I had the pleasure to work with you for a few months. So
Yeah, it was nice.
I know.
It was nice.
Yeah, it was really nice. We must repeat that.
Yeah.
And please send my best to Dr. Anna Blom, who so
Absolutely.
kindly accepted me in her lab. And
Yes.
I know this is such a great group. So it's
Thank
very
you so much.
fortunate, okay? And the audience is also congratulating you and saying that it was a brilliant presentation. So
That's good.
thank you so much.
Thank you so much. So
Okay? Have a wonderful day.
You too. Bye bye.
Bye. Bye. Thank you. Então, pessoal, muito obrigada a todos aí que assistiram, colaboraram com o nosso evento. Espero que esse seja o primeiro de vários, né? International Workshops que a gente tenha, com convidados aí de diversos países. Obrigada a todos pelo apoio, são vocês que tornam o evento possível, né? Vocês que, a audiência, as perguntas que vocês fazem. E queria deixar o convite, né, que a Serena colocou no final aí. Então, se alguém tiver interessado aí na vaga de pós-doutoramento, o laboratório é sensacional, as instalações são excelentes, né? Um laboratório de ponta lá na Suécia. Então, quem tiver interesse, pode entrar em contato comigo e eu posso ajudar nessa ponte. Tá bom? Queria agradecer a todos novamente pela presença no evento, né? E dizer que os certificados vão ser disponibilizados conforme a inscrição de vocês lá no nosso site do evento. Tá bom? Então, muito obrigada a todos e um ótimo dia.